And now we get to that combination of mitosis and cancer, where the two things we're studying at the moment come together. And it's sort of looking at where the checkpoints in the cell cycle start to control whether or not new cells will be produced. That is, whether or not mitosis will go ahead. And if mitosis does go ahead, it might be absolutely wonderful for the organism because it's repairing damage or making new cells in a growing area. Or it might be absolutely disastrous for the organism because the uncontrolled cell division might lead to cancers, tumors, malignancies. And so let's have a look at how these all fit together. And the first thing that we have to look at is a metabolic pathway involving these three substances. So firstly, cyclins. Now I'll come to this graph in a minute, but essentially you can see that there's four different classes of cyclins that wander about in the cell during its progress through the cell cycle. Now normally, this first one, which just happens to be called cyclin D, is at a very low level. And if it's at a very low level, then the cell stays at G0. And G0 is like an indefinite gap phase. It will not go on to S phase or obviously G2 or mitosis. And so until the cyclins start to appear, nothing happens in terms of cell division. The cell just stays continually expressing its genes, producing what it produces, and doing what healthy cells do. However, when something happens, when a set of ligands come and attach to some receptors on the outside to say the cell density is too low, or there are insufficient cells producing protein X, then that's when the level of cyclin D might begin. You'll notice there's four classes of cyclins, D, E, A, and B, and that they all reach peaks at different points during the cell cycle. And they only reach those peaks if the cell cycle is to go ahead. And that's because the cyclins, although they are dependent on the level of ligands that come and attach to specific receptors in response to certain environmental conditions in the cell, they control the rest of the process. So cyclins, if they're present at certain concentrations, certain critically high concentrations, they activate CDKs, and they are cyclin dependent, well obviously because they're dependent on cyclins, kinases. Cyclin dependent kinases. And a kinase, as you know, can phosphorylate another protein or dephosphorylate it. And generally, if it phosphorylates it, it provides it with a certain energy supply which allows it to go further down the line and provide energy to another protein. And what it does with these proteins, these maturation promotion factors, these are another class of many, many different kinds of proteins that will then allow the DNA to be expressed. So it might remove a blockage from a DNA molecule, and in removing that blockage, it allows, say, some DNA polymerase to come along in, or some RNA polymerase to start transcribing the gene in question onto a messenger RNA molecule, which then goes out of the nucleus and begins to construct a protein on the protein synthesis phase that happens on the ribosome. So here's the three things that are important. Firstly, the cyclins, the level of cyclins dependent on molecules coming in from the environment. Then the cyclins activate CDKs. CDKs phosphorylate something else, a maturation promotion factor, or you might think of it as mitosis promotion factor, because in effect, that's what it is. Now, essentially, when you look at the cell cycle, here it is, G1, S, G2, and then the phases of mitosis, prophase, prometaphase, metaphase, anaphase, telophase, blah, blah, blah. Three checkpoints at which the cyclins must be at exactly the required concentrations in order to activate the cyclin-dependent kinases, which can then activate the maturation promotion factors that unblock particular genes on the DNA to allow the proteins to produce that will let the next stage happen. And so we've got a checkpoint around about here at the end of G1. And this basically converts G0 into go. And then as soon as the cell enters the S phase, then we can see that that is when mitosis is going to start. There's another checkpoint at the end of G2. So if the cell has not managed to make enough organelles, if the cell is not viable, and if it does divide, then those cells will be unable to survive or unable to carry out their function, then no. That checkpoint says no go, and it does not activate the CDKs that will be necessary for the next phase to happen. And the last one occurs at the end of metaphase and does not allow anaphase to happen. And this happens if not all of the centromeres of the chromosomes are attached to their relevant kinetochore fiber. If there is a kinetochore fiber that's not attached to a centromere, or in fact the kinetochore on the outside of the centromere, then the CDK is not activated, the maturation promotion factor does not unblock the gene to allow the next stage to happen. And so, what we have is four particular and the way they interact with different CDKs is extremely complex, and it is the subject of an enormous amount of research. But we can see that when D is at a high level and is joined by a high concentration of cyclin E, then that allows us to pass the next checkpoint. It allows us to pass the G1 checkpoint and enter synthesis. And now the DNA can be replicated. Awesome. And then to pass on to the next checkpoint at G2, to finish that one, we've got to have a critical high level of A and B together. And you'll notice it's a fail-safe thing. You've got to have both of them because we don't want these cells replicating when it's inapplicable, because then the likelihood of getting a tumor is much higher. And then finally, this B, its stage here, it must be high and the others must be low in order for mitosis to continue, for metaphase to start going into anaphase as those chromosomes are pulled apart. So, going back to what this does, and it's massively more complex than I've said it. It depends on the relative concentrations of a whole bunch of proteins within the cell. But essentially, whether a cell goes forward and carries out mitosis is dependent on level of cyclins. And these levels of the four different classes of cyclins interact to promote some or activate some CDKs. And once the CDKs are activated, because they are kinases, they can then phosphorylate certain molecules, which will then allow the genes to be unblocked. And so the next set of proteins or the next set of enzymes can join on to the DNA to carry out the whole cell cycle. It's quite good. It's a really good fail-safe system. It generally works. But when it doesn't, is when we start seeing cancers.